This webinar is going to cover the anatomy of the nephron. The nephron is the functional unit of the kidneys, and you have millions of nephrons in each of your kidneys. Now, as a reminder, blood is entering the kidneys via the renal arteries, and then you go segmental arteries, interlobar arteries, and arcuate, etc., etc. Eventually, that blood's going to make its way up into the cortex where we have the glomerulus, and we're going to have filtration. Now, the glomerulus is just one part of our nephron. The filtration, we're going to be filtering out good substances as well as bad substances from the blood. We want to get rid of waste products, but we're also going to be filtering other substances that we may need to recapture. So another process that will take place in the nephron is something called reabsorption. So the nephron tubule, you're going to be responsible for reabsorbing any good things and eventually go back into your bloodstream. But we also have a process of secretion where substances are going from the blood, the peritubular capillaries, into the nephron tubule. And so the ultimate is whatever you filter, you secrete, and then minus anything you reabsorb is what's going to end up being excreted. That'll be your urine. So I want to show you there are actually two different types of nephrons in the kidneys. We have what referred to as cortical nephrons, because you see the majority of that nephron is found in the renal cortex. Most of the nephrons in your kidney are cortical nephrons. They're going to be responsible for primarily getting rid of those waste products. You'll notice is down in here, not very much of it, if at all, enters that renal medulla. But juxtamedullary nephrons, because it's kind of juxtaposed next to the medulla, the juxtamedullary nephrons, you'll see parts of it out here in the cortex, but you see this structure here called the loop of Henle, very long loops of Henle that go deep down into the medulla. They're going to be very important in water balance, so we're going to be looking at that at a later time. Now, just as a reminder, is the urine is going to be formed in these nephron tubules, and eventually the urine is going to be traveling down in this direction here, down and deep into the medulla through these little collecting ducts, and eventually down in this region here. This is where your renal papilla is. And that urine goes from the renal papilla to your minor calyx, to your major calyx, renal pelvis, ureters, and you can go on from there. So what I'm going to do is I'm going to show you, in the process of showing you the different parts of the nephron tubule, I'm going to show some models so it makes it a little bit easier when you go to lab. You'll see here, this is a cortical nephron. See most of it, and this is like all of it's up in the cortex. This one here is primarily a cortical nephron. This one here is a juxtamedullary nephron. Very, very long loop of Henle. Now, as a reminder, like here, this would be referred to as the vasa recta. The blood vessels up here, you see here, are going to be referred to, as you see here and here and here, are peritubular capillaries. Now, there's two major components of our nephron. The first one is referred to as the renal corpuscle. Now, the renal corpuscle includes, if you look right up here at these, that red is the glomerulus, and then you see the yellow surrounding it is Bowman's capsule. So that's the renal corpuscle. The renal corpuscle, this is where filtration takes place. So on these ones, you see they haven't cut it. But in these ones, they cut it so you can kind of see in there. So those collectively are the renal corpuscle, and I'll show you another way. So the renal corpuscle is what you see here. Here is Bowman's capsule, and here is your glomerulus. So that's your renal corpuscle. The filtration will take place here. So substances will leave, mainly driven by blood pressure, and enter the Bowman's capsule. And filtration is very high pressure. And pretty much everything leaves the blood except blood cells, large proteins, or anything bound to a protein, just because the way this is set up. Those fenestrated capillaries, in the way that the Bowman's capsule is designed is that you have, it's relatively non-selective filtration, but you have large substances that cannot be filtered by the blood. The other component of our nephron is, besides the Bowman's or renal corpuscle, is the renal tubule. So all the rest of this is the renal tubule. And that's what we're going to be looking at. We're going to be looking at the different components of the renal tubule. And periodically I'll talk about some major things that take place there at each part. So you have this picture on the left in your notes, and so we're going to kind of go along and show you the different parts. Now the first part you see here is the proximal convoluted tubule. It's convoluted, it's kind of twisty and turny. Proximal, because we're proximal, think of it as its directional term to where that glomerulus is, okay, where filtration took place. Because we're going to look at something later called the distal tubule. So you have a proximal convoluted tubule, and any part where it's no longer convoluted is just the proximal tubule. I don't care if you say proximal tubule versus proximal convoluted tubule, that really doesn't bother me. The proximal tubule is special in that it has a lot of microvilli, and this is a part of the nephron tubule where a lot of reabsorption takes place. Majority of your water and your electrolytes are going to be reabsorbed here. This is where all nutrient absorption takes place. So I'm talking about glucose and fructose and amino acids. That's the only place where those things are reabsorbed. The histology of that part of the nephron tubule will show you, if you look at it, they're like kind of, they're cuboidal cells, they're fat, they have a lot of microvilli, is it has to have a lot of transport proteins for the processes of reabsorption. So there are a lot of reabsorption that takes place in that proximal tubule. Then we come down into what we refer to as the loop of Henle. And the loop of Henle has more than one component to it. So as we're moving down this way, that's referred to as the descending limb of the loop of Henle, because we're descending. It's traveling down towards the renal pelvis. And then we're going to start coming up. In this first part, they refer to as the thin ascending limb of the loop of Henle. And then we see here, this is the thick ascending limb of the loop of Henle. And you must distinguish between the thin and thick segments of the ascending limb of the loop of Henle, because there's different things that take place there. And the reason why you have thin versus thick, if you look at the descending limb of the loop of Henle, it's thinner than any other parts of the nephron tubules besides that thin ascending limb. The cells are more squamous cells. The only thing that takes place in the descending limb is reabsorption of water. So you don't have to have a lot of bigger cells for transport proteins, because it's just reabsorption of water. The thin ascending limb, not a heck of a lot goes on there. I usually kind of ignore it, but you do have some movement of some passive movement of some sodium, some urea, and a little bit of water. Now, the thick ascending limb is very important in that this part of the nephron tubule is impermeable to water. So water does not move across the membrane at all, but you have reabsorption of electrolytes like sodium and potassium and chloride and calcium. So when the contents are moving up and you get reabsorption of solutes, but water stays behind, is that the contents become diluted. Now as you look, if I go back here to the descending limb, because there's only reabsorption of water but no movement of solutes, is the contents of the tubular fluid as they move down become very concentrated. And we'll talk about the significance of what's going on in the loop of Henle at a later date, but it's going to be very important in creating an environment in which we'll be able to, under certain circumstances, be able to produce a very concentrated urine. Now if we kind of keep moving up the nephron tubule, you'll notice right here, this part here is called the early distal tubule. So when we start talking about distal tubule, there's three different sections I'm going to talk about, and you must distinguish between them because there's some differences. Now, the early distal tubule does similar things as the thick ascending limb of the loop of Henle, but there's one other thing that's going to be important that you know about the early distal tubule. On this picture here in this model, this right here is part of the early distal tubule. And get my eraser. I'll get my eraser. There's cells in the early distal tubule that are referred to as macula densa cells. They're in physical contact with cells of the afferent arteriole. So here, this is right here, this is the afferent arteriole. And the cells here are called juxtaglomerular cells. The cells here that are in contact with those juxtaglomerular cells are macula densa cells. Collectively, that makes up what we call the juxtaglomerular apparatus. That structure is its function is regulation of GFR, which is glomerular filtration rate. So the juxtaglomerular apparatus, its role is in regulation of glomerular filtration rate. So it's very important that you know that. Now if I look here, actually I gotta come back here. I think I had another picture. So I'll come back here. So you see again, macula densa cells, juxtaglomerular cells, collectively juxtaglomerular apparatus. Now the juxtaglomerular cells, where it releases renin, which was important in regulation of blood pressure, but that's also going to be very important in regulation of glomerular filtration rate. Those cells also produce erythropoietin. Now, erythropoietin doesn't play a role in regulation of glomerular filtration rate, though. But erythropoietin is secreted from those cells in response to hypoxia, and that will be important in regulating blood cell production, or red blood cell production. Now, as we move along the nephron tubule, you see where the tubule becomes convoluted again. So that's the distal convoluted tubule, which you can abbreviate like that. Proximal convoluted tubule, you may abbreviate like that. The other parts, you can't abbreviate, okay? So if I abbreviate it, it's perfectly acceptable. If I do not abbreviate it, you may not. Now, the distal convoluted tubule does have absorption of electrolytes, secretion of things like maybe hydrogen ions. So the distal convoluted tubule is going to be involved in pretty much movement of more ions, electrolytes. Now but we go later, right in here, that's the late distal tubule. That's important structuring, and it's different from the distal convoluted tubule in what it does and what it's responsive to. There's two different types of cells in the distal convoluted tubule. So the first cells I want to talk about are called principal cells. And this is the spelling, because there's more than one way to spell principal. So principal cells are involved in reabsorption of sodium and secretion of potassium. And these are cells that are responsive to the hormone aldosterone. So when you need to, say, get rid of excess potassium, aldosterone can be secreted by the adrenal cortex and target those principal cells. Okay, so principal cells are involved in reabsorption of sodium and secretion of potassium. The other type of cells are called intercalated cells, and there's actually more than one type of intercalated cells, but we're just going to make it simple right now. These intercalated cells will be important when we talk about acid-base balance. They're involved in secretion of the hydrogen ions, reabsorption of bicarb, but depending on what the type is and what's going on in your body, we may not need to secrete hydrogen ions, we may not need to reabsorb bicarb, but we're just going to say these are very important in acid-base balance. The late distal tubule is also a target for the hormone ADH. So the permeability of this part of the nephron tubule is dependent upon the levels of antidiuretic hormone. ADH increases the permeability of this part of the nephron tubule to water. Prior to this, the only parts of the nephron tubule that were freely permeable to water were proximal convoluted tubule, the descending limb of the loop of Henle, and we're just going to ignore the thin ascending limb. But thick ascending limb, early distal tubule, distal convoluted tubule is not permeable to water. Now, as we keep going on, actually I got to go back here, is then you see here, this part is called the cortical collecting tubule. Found up in the cortex, it's collecting fluid from various nephrons. So you got, this would be a late distal tubule coming from another nephron. Here's another one. So they're collecting urine from various structures, and it's found in the cortex. And the cortical collecting tubule has the same cells as the late distal tubule. So it has intercalated cells. It has principal cells. It's responsive to aldosterone. It's responsive to ADH also. Now then, as we keep moving down, once you go into the medulla, it's referred to as the medullary duct. And the medullary duct is going to have reabsorption of electrolytes. It's also, the medullary duct its permeability to water is also dependent upon the levels of anti-diuretic hormone, so ADH. It's also permeable to urea. And as the levels of ADH increase, the permeability to urea increases, and it seems kind of odd because urea is a waste product, but when we talk about conditions and where I need to be able to excrete a very concentrated urine, I need to conserve water, urea is going to play an important role in this whole process. So I do want some urea be able to leave this tubule, but it's going to stay in the interstitium, okay? And we'll look at that at a later point. What I want to do is I want to just use some models, lab models. So if you don't want to continue on, you can stop, but this will help you in lab, is kind of show you really quickly some of the parts of the nephron tubule that I had mentioned. So let's look here. So we got proximal convoluted tubule, proximal tubule, descending limb of the loop of Henle, and this doesn't show you thin ascending very well, but so here's thick ascending limb. There's early distal tubule, right there. Distal convoluted tubule, late distal tubule, cortical collecting tubule, medullary duct, and then when we get down in here, these are when we get down to the papilla, we refer to them as papillary ducts. Okay, so you see it one like that. Now this one over here, you can see that the thin ascending limb better. So I'll follow this one in blue. So we have proximal convoluted tubule. Here's descending limb of the loop of Henle, thin ascending limb of the loop of Henle, thick ascending limb, and then we can go on from there. So you got distal convoluted tubule, so I'll just stop there because I may go on forever. Now this is another model that you'll need to look at in lab, and so it seems a little odd, but it's very good at showing you different parts of the nephron tubule as well as some blood vessels. So on this model, from here to here represents the medulla. Up here is the cortex. And so what you'll see, at least here, these are arcuate arteries. This would be interlobular arteries. And you'll notice here, this is an afferent arterial, this is an efferent arterial. Way you know it's the efferent arterial is that forms the peritubular capillary system. Now here, these down here are vasorecta because we're deep down into the medulla. Up here, these are peritubular capillaries, okay? And then let's erase this so we can look at some parts of the nephron tubule. And then we'll zoom in a little bit more. So what you'll see, let's focus in on this, is you have in the orange is the proximal convoluted tubule, okay? And then here's the proximal tubule right in there. Then you see in the purple, descending limb of loop Henle, and I'm coloring it over in orange. Then I'm coming up thin ascending limb, thick ascending limb, distal convoluted tubule, the early distal tubule will be right up in here, there. Then you see in yellow, the little bit in yellow, is the late distal tubule. Then we kind of come down, cortical collecting tubule, medullary duct, and then down in here we got papillary duct. So let's zoom in a little bit. So we're up in the cortex. So again, you see the proximal convoluted tubule in the orange right here. And the purple is the distal convoluted tubule. There is the late distal tubule there. On this one, let's look way up on this one, see if we can see. So they kept the colors the same, so make it easier on you. So the orange is the proximal convoluted tubule. You have in purple up here is the distal convoluted tubule. You've got proximal tubule here, descending limb of loop of Henle, and they didn't do a good job of thin ascending limb, so we can't really show it there. But thick ascending limb of loop of Henle, early distal tubule, so you can kind of see it from there. And then here we've zoomed in down at the bottom again, so you can kind of see some of what we have there. So make sure you look at these in lab, okay? So I'm just going to stop here.